This podcast is for the second year course, Genetics. The course is offered in the autumn term and is worth 12 credits. The course has 3 types of regular meetings. There are 3 hours of lecture per week for a total of 27 lectures over the 10-week term. The lectures are the primary source of course material and will provide the framework for other aspects of the course. There are 4 one-hour tutorials that meet in weeks 3, 5, 7, and 9. The tutorials will expand upon the lecture material and provide an environment for problem solving and discussion. There is a laboratory practical consisting of 3 4-hour sessions that include 1 hour of pre-lab and 3 hours of lab time. There will also be a post-lab meeting during week 8 to offer advice on data interpretation and writing up of the practical report. Outside of these meetings, students can meet up with the lecturer or their course tutor during office hours or by appointment. Genetics has 5 assessments. The coursework includes 3 problem sets that are due on the Thursday of weeks 3, 5, and 7, each of which is worth 10% of the final mark on the course. The problem sets are designed to test the students' understanding of the course material and develop problem-solving and hypothesis testing skills. Problem sets are returned to the students during the term and thus provide immediate feedback on their progress in the course and their understanding of the course material. The second type of coursework is the practical report, which is due on the Thursday of week 10 and is worth 20% of the final mark on the course. The practical report is, in principle, an extended problem set that students complete using data they have generated during the practical sessions. The report is written up in the format of a scientific paper, and students are marked on how they present and interpret their data. The last assessment is an end-of-year exam, which will take place in June. The exam is worth 50% of the final mark for the course, and in format it is similar to the problem sets. The textbook that will be used for the course is the 3rd edition of Genetics, a Conceptual Approach by Benjamin A. Pierce. The book is available from W.H. Freeman, can be purchased from Amazon, and will be on sale at the University bookstore at the start of term. The genetics course is taught assuming the students have no previous experience in the subject. As such, there are no prerequisites for the course. It helps if you have taken fundamentals of cell and molecular biology and introduction to molecular genetics, but these courses are not essential to do well in genetics. Genetics is a prerequisite for the 3rd year course, Genes and Development. The course will cover material on all aspects of eukaryotic genetics, that is, the genetics of plants, animals, and fungi. This material falls into 10 broad sections. Mendelian genetics includes the basic rules of heredity as discovered by Mendel, and this section will also include probability and how probability is used to predict the inheritance of genes. Mitosis and meiosis cover the biology of cell division, and in particular how gametes are formed by meiosis, which is directly related to the rules of heredity that were discovered by Mendel. The section on sex linkage brings the first 2 sections together by linking Mendel's rules of heredity with the behavior of chromosomes. Despite its name, sex linkage is a special case of simple segregation and is discussed in this context. In this section, the phenomena of non-disjunction will also be discussed. The longest section of the course is linkage and recombination, which recovers the genetics and biology of the exchange of genetic material between homologous chromosomes during meiosis. This section also includes the related topics of recombination mapping and the making of genetic maps. Mutation in alleles covers how alterations in the genetic material arise and how this affects gene function and, as a consequence, phenotype. This section will include complementation, different allelic states, extensions to dominance, and the genetics of lethal and conditional mutations. The section on chromosomal aberrations will cover all mutations that affect the organization and number of chromosomes. This includes deficiencies, inversions, translocations, duplications, aneuploidy, and polyploidy. Human genetics covers how the basic rules of heredity are applied to humans and will include special techniques that are used to analyze the inheritance of traits associated with human disease. In gene interaction, the fundamental aspects of interaction between phenotypes is presented, and how this kind of information is used to understand genetic pathways that regulate development. The section on transposable elements covers the origin, behavior, and genetics of mobile genetic elements and their effects on genomes and genome evolution. The final section, on non-nuclear inheritance, covers the genetics of organellar genomes and how this relates to the inheritance patterns of the nuclear genome. In principle, genetics is a formalization of the relationship between an organism's genes and its morphology, physiology, and behavior, or, in genetic terms, between its genotype and its phenotype. In this light, genetics is clearly one of the fundamental elements in biological science, as it links the studies of genes and cells, what we learn about in biochemistry and molecular biology, with the studies of the organism, its fitness, and its interaction with the environment, what we learn about in behavior, evolution, and ecology. The study of genetics leads to an understanding of genes and their functions, the patterns of inheritance of genes from one generation to the next, the interactions among genes and between genes and the environment, and the role that genes play in selection and fitness of the individual for its environment. Thus, the concepts of genetics touch upon every discipline in biological science and form one of the core elements in biological thought. If you have further questions about the genetics course, you should contact the course organizer and lecturer, Dr. Robert Ray. Dr. Ray can be found in room 4C10 in the John Maynard Smith building and can be reached by email at r.ray@sussex.ac.uk.